Hello and welcome to this video building on our knowledge of sugars. Sugar is necessary for brewing as it is the only way to get alcohol and energy for your yeast. There are two sugars that we continue to mention, alpha glucose and beta glucose. We have spent so much time on these two as they are the foundation to starches and cellulose. There is a simple distinction between them. An alpha-glucose molecule has the hydroxyl group pointing upwards. A beta-glucose molecule has the hydroxyl group pointing downwards. A simple distinction for sure, but it covers the key point. Glucose is the simplest example of an alpha and beta sugar. They make starch and cellulose respectively. This difference in structure makes it impossible for the same enzymes to break down each structure the same way. We previously described the two major forms, cellulose and starch, in greater detail. What is important in this is the way each is formed and functions when mashing grains and in other applications, like ethanol production for fuel, which can be done with the use of cellulose. Making the most of starch is essential for grain as it is growing. We want to make the most of starch to get the most glucose, which will provide the best value for brewers. In order to get glucose from starch, we need to convert it. This is generally done using barley and a number of enzymes, two of which we've described in the past, alpha-amylase and beta-amylase. Both of these function differently, but in similar ways. They break down the long, complex chain of starches into short sections of glucose, which can be metabolized by yeast. Alpha-amylase breaks the bonds at random points. This creates short, linear sections that can be worked on by the beta-amylase. You can, to a certain extent, control how effectively this occurs by altering the time and temperature at which you are mashing your grains. By changing the time the alpha-amylase has to work and the beta-amylase has to work, you can more effectively extract larger amounts of sugars that can be brewed with. This can be improved further with the addition of other adjuncts. There are a wide range of temperatures that are relevant to the brewer when they are doing this. If you are taking unmalted grains, you want to extract as much as you can at a temperature around 40 degrees centigrade. That is the ideal temperature range for these enzymes to function, both the beta and the alpha. Then, when you want to start removing all the branches, you raise that temperature slightly. This keeps going until you eventually get to a point where you have extracted everything you can from the unmalted grain. But that's not as commonly done. More often than not, we are trying to extract glucose and similar compounds out of malt. And malt is a little more difficult. Saccharification starts between 55 and 66 degrees centigrade. More often than not, brewers will bring their initial boil up to 50 degrees, maybe 55, and then gradually take it into a temperature around 60. The reason they try and hold it around 60 degrees centigrade is that it will produce the most amount of usable sugars, as it is the ideal temperature for beta-amylases. This will then gradually be raised again, taking it up to 60 to 63 degrees centigrade. This is where all of the residual sugars that can be extracted will be extracted. The temperature is then raised again, which helps to partially denature the beta-amylase, but then also allow for the alpha-amylase to begin working the way it's meant to work. This is the ideal range for alpha-amylase. From here, it's then raised higher yet again. it goes to 65 degrees and then up to 68 and then 70 degrees. Somewhere around this 70-degree mark, you have the amylases achieving peak efficiency. This will give you your maltose, your glucose, and a few other saccharides. Then you push it past 70 degrees, all the way up to 76. At 76 degrees, all of the enzymes have become denatured. You have broken down as many of the long starch chains as possible, and then you have further dismantled them into shorter fermentable sections of saccharides. Knowing this, you can begin to imagine there are reasons why different beers taste different. Other than the obvious use of differing yeast strains, the nature of the malt used, but also how it has been mashed. Drier, clearer, less bodied beer with a higher alcohol content is generally made through a more extended mashing process. This will give it a higher gravity initially, but a lower final gravity in the end. This is done by increasing the temperature space between the beta and alpha amylases and holding them at those temperatures for longer. This will ensure a better breakdown of the total starch chain and then the short sections to get those fermentables. If, on the other hand, you are after a full-bodied, sweeter tasting beer, then you want to decrease the amount of time that you hold it between those different enzymes being active. The more time the alpha amylase and then the beta amylase have to act independently of each other increases the amount that can be converted into ethanol in the end. This will, of course, also decrease the ethanol concentration in your final beer product. The right temperature, the right time, and taking advantage of the right enzyme is how you get different results. This is something we can see with different beer styles. As a general rule, the way a lager is made is very different to an ale as far as choice of malt and mashing is concerned. It leads to a far cleaner taste, a slightly higher alcohol content, and a very different product. Compare this to an ale, which is generally much sweeter and has a fuller body. This alone should tell you a lot, but it becomes more complex when you look at different grains by region. And yes, you can become region locked. This is influenced by the nature and stability of the amylases, the nature and to a certain extent complexity of the starches involved, and the ability to account for these in what you are working with. Amylase stability is incredibly important for the temperature-controlled conversion of starch to glucose. We'll go into this in greater detail eventually, but for this video, understand that if you have a grain sourced from a region where the amylase stability is low, you need to control and have a much more specific temperature range that is held for a longer period to get the best extraction. Conversely, highly stable amylases may give you greater flexibility in what you do, allowing for more crossover. Depending on what you are trying to brew, this could be a very relevant factor to allow for. Then there is the grain itself. Regional variations in grain can also affect the degree of complexity of the starch and everything else in there. Is there going to be a higher protein concentration, which will also influence the effective extraction of your saccharides, and a variety of other factors that vary massively by region and by strain of grain being extracted from. Even within barley, there is a considered range of content and what percentage or concentration each grain granule has. All of this is important to keep in mind when brewing region-specific beers, when you are using a regional malt or grain variety that is local to you but not necessarily to the style. They will likely to be affected at a subtle and low level The means you've used to mash can amplify this factor considerably, which means what was once subtle and low level becomes pronounced and at the surface of what you are brewing. Thank you for watching this video. Please consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.